Thank you. Thank you all for joining. I'm Neha, and it's my pleasure to introduce Dr. Slavov as our speaker today. Dr. Slavov received his undergrad education from Massachusetts Institute of Technology in 2004 and holds a PhD from Princeton University for characterizing the coordination of cellular growth with cell expression and metabolism. He's the founding director and CEO of Parallel Square Technology Institute and currently a professor at Northeastern University. His lab pioneered experimental and computational methods for single-cell proteomics and used them to connect protein covariation across single cells to functional phenotypes, including macrophage polarization, emergence of drug-resistant priming, early mammalian development, and stem cell differentiation. Most recently, the Slavov lab developed methods for high-throughput single-cell proteomics by mass spectrometry and used them to quantify proteome heterogeneity during cell differentiation. Dr. Slavov also organizes the annual single-cell proteomics conference every year, happening this year on May 27th and 28th in Boston, if anybody wants to join. So without further ado, please welcome Dr. Slavov.
Thank you very much for the kind introduction. I'm happy to be able to join you, at least remotely, and I wish I could join you in person. So the plan for today is to share with you some of our recent results on studying the proteome, and I will in particular share an observation that has really changed the way I think of the human proteome. And then I will give you a quick conceptual overview of single-cell proteomic technologies and one example of how we can use those technologies to measure protein variation across single cells from which we can then infer regulatory mechanisms. So when we think of the proteome, it's important to recognize scale and complexity. There order thousands of messenger RNAs in a single human cell and order billions of protein molecules. And they're not just thousands-fold more protein molecules, but they're also more varied in terms of modifications, interactions, and so on. And we normally think of protein abundances being in large effect determined by rate of protein synthesis. But of course, there is another side of this equation, which is the rate of protein degradation or protein clearance. And recently, by analyzing protein abundance variation together with direct measurements of protein synthesis and degradation rates, we found that protein degradation variation contributes about as much to protein abundance differences across cell types as the synthesis side of the equation, as long as the cells are not rapidly growing. In rapidly growing cells, such as in vitro cancer cell lines, much of the protein removal happens by dilution rather than degradation, and there the effect of protein degradation is smaller on protein abundance variation. So there is this important side of the protein degradation. But even when we think of the synthesis side, despite that being the focus of much more intensive studies, it remains incomplete in some ways and indirect. Specifically, what are the sequences of the proteins that make up our bodies? It has been too challenging for us. We have not yet developed technologies capable of determining protein sequences at the scale of a mammalian proteome, and that's why we indirectly predict those sequences from nucleic acids. And about in 1960s, about 60 years ago, this young man, Marshall Nirenberg, working in the intramural wing of NIH, came up with a clever idea to use synthetic RNAs translated in vitro by E. coli cell extracts, from which he was able to deduce the genetic code. And this is the mechanism that we use to predict the protein sequences in our bodies. which has been remarkably successful. It has been applied to a variety of different organisms, but we haven't had the opportunity to really examine what are the sequences of proteins beyond those predictions. There, we're not testing other hypotheses. We know that protein synthesis is not a perfect process, certainly from using artificial genetic constructs, reporter systems. Colleagues have estimated that rates of incorporating amino acids deviating from the genetic code is a few percent in some cases, but those are artificial reporter constructs. We were very interested to be able to see to what extent those deviations happen with endogenous human proteins in the human body. So to this end, we exploited the fact that if there is an amino acid substitution in a protein, in a peptide, in a protein, that is going to alter the mass spectra in a very predictable way. And using this expectation, we analyze billions of mass spectra and billions of Illumina reads from over 1,000 human patients from different tissues, both healthy tissues and from cancer patients. We use the RNA sequencing data to first in silico reconstruct. Can you hear me? There is some noise. To first in silico reconstruct transcriptomes, having about 70 transcript isoforms, 70,000 full-length transcript isoforms predicted per patient. And then we looked for the corresponding protein products, and we found that there are millions of modified peptides, and many of those modifications correspond to known post-translational modifications, whether biological or artifactual during sample preparation. Some of them originate from sequences of the transcriptome designated as non-coding. But we were particularly interested and focused on variations in sequences where the incorporated amino acid in the protein differs from the one predicted by the genetic code. And we found highly confidently large number of such changes in peptide sequences. And we compared the abundance of the protein form, the proteoform containing the protein sequence predicted by the genetic code, to that containing the substituted sequence. And surprisingly, for many proteoforms, the substituted version appears to be more abundant, and this includes many transcription factors, many signaling proteins, many enzymes, and so on. So it really spans the gamut of functions for the proteins. And we were very surprised by this observation, not only that we have these alternate protein products, but they appear to be highly abundant products of alternate RNA decoding. So we were very skeptical. We looked at the raw data, and here is an example where we have detected 86 reads that perfectly map to the genome corresponding to the canonical form. Not a single, and we also detect the corresponding peptide sequence with high confidence predicted by the genetic code. But interestingly, we also see another peptide sequence that instead of having here asparagine has alanine. And this is just as confidently identified. This is the more abundant peptide, 16-fold more abundant, and there isn't a single RNA read coding for this particular peptide. So that's interesting, but that's an individual example. So we also used deep learning to explore much more systematically this type of analysis. And in particular, deep learning methods have made it possible to accurately predict peptide properties such as fragmentation spectra, retention times, and so on. So we can use such predictions to update our statistical confidence in the identified peptide sequences. And on average, when we did the deep learning risk scoring, we found that we have 10-fold higher confidence than from the original identification. So this gave us even more confidence that these peptides really exist in the human body. We also wanted to make sure that our quantification is reliable. So to this end, we used peptides produced from different proteolytic enzymes that digest the same proteins but produce different peptides. And we look for a particular section of the protein sequence if the estimated frequency of alternate amino acid incorporation is consistent across proteases. And we see that the consistency is quite good. When we use different enzymes to digest the proteins, we produce different peptides, we estimate the same rate of amino acid incorporation deviating from the genetic code. So what's causing this? One side, of course, is the synthesis rate, and we see that the number of mismatches between the codon on the RNA and the anticodon on the tRNA has a high predictive power. Amino acid incorporations that correspond to a single mismatch have about 50-fold higher abundance than those that require multiple mismatches. We also see that there is a strong correlation with codon frequency, where less frequent codons corresponding to less abundant tRNAs have much higher rate of incorporating different amino acids. And there's also a significant correlation to nucleotide modifications, such as uridine being modified to pseudouridine. And these observations clearly indicate that there is a role of translation, of alternate translation in determining those different sequences. But there's also an important role on protein stability. And in particular, we can directly measure protein turnover and rate of protein synthesis and degradation by metabolic pulse of isotopically labeled amino acids. And here you can see an example where we measure the turnover rate for, in blue, the peptide predicted by the genetic code, which is relatively low, and the turnover rate in orange to the alternate RNA decoding, and this is much higher. And this is consistent with some of these alternate RNA decodings resulting in highly stable protein products that then accumulate because their degradation rate is low. And quite naturally, one can expect that with age, the abundance of such stable protein products will increase, and some very, very preliminary evidence suggests that this indeed is the case, and it seems to be associated with neurodegenerative diseases, but this is very preliminary and clearly needs more focused systematic studies. We were able to identify a number of predictors for factors for the rate of amino acid incorporation deviating from the genetic code, and to predict with good accuracy their occurrence. And those predictors include the level of conservation of proteins, protein disorder, and so on. And there's a lot more interesting things that I can tell you about this surprising observation, but I'll just finish with this collection of observations because I would love to talk about single-cell proteomic technologies and what we are finding there. So we have found many associations for the abundance of alternately translated protein products with diseases. So for example, this particular substitution here is significantly more abundant in tumors and in the surrounding non-tumorous tissue in 3 different types of cancer. We also see systematic differences between different substitution types across different cancer types. We can also see certain signaling pathways showing high prevalence of alternately decoded protein products. And there is significant enrichment with signaling proteins. For example, there are some amino acid motifs that are associated with those. And lastly, that I'll share today is there is clustering both in the linear sequence space of proteins, where those substitutions are observed, and in the three-dimensional space of proteins. In some cases, the substitutions occur at sites that are distant in linear space, but when the protein folds, they cluster close to each other in 3D space. And now I'm going to switch gears and talk about doing single-cell protein analysis with mass spectrometry. And this involves not only measuring protein abundance, but also measuring rate of protein synthesis and degradation. As I said at the very beginning, that's very important to understand the mechanisms that regulate protein abundance variation, measuring various modifications such as phosphorylation, measuring protein complexes, and so on. So to give you a big picture view of what the technology is currently capable of doing, we have methods that allow to prepare order thousands of single cells per day. We are able to analyze between hundreds to over 1,000 single cells per day, and the higher number of analysis corresponds to multiplexing, as I'm going to talk more about in a few minutes. And generally, we count order tens of millions of protein molecules per single cell, and those correspond to order thousands of gene products, different proteins. The number of quantified proteins depends certainly on the number of cells, substantially fewer proteins in tiny T cells, many more proteins in vitro grown cells, and so on. So to give you a sense of sample preparation, that clearly was one of the important challenges to overcome for the field, going through multiple iterations. At the moment, one of the most scalable platforms is this droplet preparation on the surface of glass slides. They have the exact shape and form as microscopic glass slides, and they're covered with fluorocarbon coating to decrease surface wetting and keep them nicely shaped. And then each of these droplets that you can see here serves as the reaction vessel in which a single cell is isolated, lysed, proteins extracted, digested, labeled if labeling is desired. And then when we do multiplexing, all of the single cells that are multiplexed are part of one cluster, and this cluster is being pulled together at the end of the experiment in a fully automated manner and transferred into a multi-well plate, so that then the multi-well plate can be placed into the autosampler of an LC-MS system to do the analysis. And as we started doing this analysis, one of the influential observations for my group early on was the fact that when analyzing even a single human cell, we could detect tens of thousands of peptide-like features. Peptide-like features look like this. So all of the peptide molecules having the same sequence are going to look like this series of peaks. The first peak corresponds to molecules having no heavy atoms. The second peak corresponds to molecules having one heavy atom, and so on. So we can identify how peptide-like features look like. And then the real question is, how can we identify their sequences? If we have so many, it is challenging to analyze all of them one at a time, which is a conventional way, classical way of doing the sequence identification. And my laboratory has developed two conceptual approaches to analyze these peptide-like, the large number of peptide-like features that are highly complementary. The first approach is what I do in my daily life. I have too many things to do on my to-do list. I can do each one of them, but I cannot do all of them because I don't have time. So we developed real-time instrument control approaches to prioritize the peptides of interest, whether they come from proteins of biological interest, less abundant proteins, they correspond to post-translational modifications, and so on. And this allows to increase the rate of data completeness to over 90%. So we are able to largely avoid missing values. It's able to increase dynamic range by an order of magnitude, thereby increasing sensitivity. And the increase is particularly important for lowly abundant proteins, and also increase the overall coverage. We are quantifying about 2 times more proteins per single cell when using prioritization. It's implemented by free software, so all you need to do is change the heuristic, the logic that by which the instrument is operated to acquire the data. The second approach to dealing with having too many peptide-like features to analyze all of them one at a time is to parallelize. And this is something that unfortunately I cannot do in my daily life as much as I wish. I'm not as good at multitasking, but computers are better at that. So let me tell you what we did in that context. There have existed methods to use isotopically encoded small molecules, mass tags, to multiplex the analysis of different samples. But those methods generally sequence only one peptide at a time. And there were methods to fragment many peptides at a time, but they weren't implemented with multiplexing efficiently. So we wanted to combine parallelization both at the level of samples or single cells, and also at the level of peptide sequence identification. And if we can do this, then we can start achieving multiplicative scaling of the rate of data generation without decreasing the depth of coverage or the quantitative accuracy or the specificity that mass spectrometry is known about, without reducing the number of peptide molecules that we count. So this was the idea and turned out to work remarkably well. I'll show you a few examples of that to build intuition. So first, starting with raw data, a deep dive in how the data look like. So here you can see a peptide from a single pancreatic adenocarcinoma cell. Eluting from the chromatography, we make 3 measurements of the peptide ion, telling us how abundant it is, relative to the same peptide in a monocyte cell. And to determine the sequence of the peptides, we fragment them, and from the fragments, very different ions, we can also estimate the abundance of this peptide. So from the consistency of those 6 data points that we get here, we can determine how reliable is our estimate for the abundance of this particular peptide in those 2 single cells. And if I zoom out from the raw data and look more globally across many proteins, we see that the quantitative accuracy and precision is quite good. And in particular, one thing to emphasize here is the large dynamic range for relative quantification, where we can quantify a 1,000-fold difference in relative abundance of a protein when using this multiplexed approach. That essentially allows us to get several times more data per unit time from the same instruments at several times lower price, because most of the cost is really associated with instruments rather than the reagents. And those advances were noticed by Convergent Research and Schmidt Futures, which is a philanthropy of Eric Schmidt, and then by Ken Griffin's philanthropy. And they approached us with interest to scale up the development of this technology, to accelerate it, and ultimately provided funding for us to start a nonprofit research institute in Boston. It's called Parallel Square Technology Institute. Its primary focus is on biomedical problems, which include that need better, more sensitive, more flexible measurements, more scalable measurements of proteins. We have multiple projects in immunology, neurodegenerative diseases, but on the technology side of proteomics, we are continuing to advance this PLEX-DI approach that I introduced to you. And in particular, we are making small molecules that can be used as barcodes for this multiplexing that not only allow to analyze more single cells per unit time, but also allow us to quantify more proteins more sensitively by stabilizing the fragment ions and therefore enabling sequencing with higher confidence. And this is particularly important when we have post-translational modifications such as phosphorylation to be able to localize it. It's particularly important when we have alternate RNA decoding, as I told you in the first section of my talk, so that we can have high confidence in surprising sequences, and very important if we wanted to do de novo sequencing. If we have a complete set of peptide fragments, we can de novo sequence with high confidence. We don't need all these hypotheses from the transcriptome that may or may not correspond to the most abundant proteoforms and the most functionally significant ones. So here is a result that we can share of a particular molecule that we have developed that give us substantially more fragments for the labeled peptides and therefore higher sensitivity and allows identifying more peptides. So this is, to my knowledge, the first example where labeling is beginning not only to confer benefit for throughput, but really conferring benefit for sensitivity, sequence identification, and so on. And we are going to have a one-day symposium on April 22nd to showcase more technology developed by PTI, by Parallel Square Technology Institute. And in the final research segment of my talk today, I want to share with you an example of how single-cell protein measurements can identify mechanisms of regulation. And first, starting with a concept, the idea is that we can think of each cell, the proteome of that cell, is representing a particular configuration, a particular perturbation. And if we have an ensemble of single-cell proteomes, we can test if different regulatory models are consistent with the data as described in these articles here referenced at the bottom. And we applied this idea to study protein transport, which proteins regulate protein transport in macrophages upon LPS stimulation. And to this end, we measured the proteins in large number of nuclei, so over 3,000 single nuclei from macrophage cells, and we were able to quantify well over 1,000 proteins. When we do these proteomic measurements, we like to benchmark quantitative accuracy with what is effectively a ginormous spike-in experiment, where we mix yeast proteomes with human proteomes in known ratios, and we determine if the mixing ratios correspond to the measured ones. And the fact that they line up here so well indicates that the accuracy of these measurements is very high. What did we find? So here's an example of a protein, which is a subunit of the nucleopore complex that correlates positively with protein transport, which means that macrophage cells that had more of this nucleopore subunit had more protein transport upon LPS stimulation. And here is the converse example, where macrophage cells that had more of the Vim protein exhibited less protein transport upon LPS stimulation. So let's see what are we learning from these data. Now, it turns out that the nuclear pore subunit is not an isolated one. We see that most nucleopore subunits correlate positively to transport, which directly suggests that protein transport to the nucleus is kinetically limited by the number of nucleopores. And particularly interestingly, we find that these correlations actually correlate to the spatial distribution, to the exact location of each subunit in the pore complex, so that the subunits that are part of the inner ring tend to be more important for transport. They have higher correlations than those from the outer ring, which is also associated with their degradation rate. So here is an example where protein variation across single cells, and correlation to transport is really suggesting how a protein complex functions in those cells. In addition to the nucleopore complex, we found a large number of other proteins that are strongly associated, strongly predictive of protein transport, suggesting that they might regulate it. And we wanted to test these hypotheses. So we used siRNAs to knock down systematically many of these proteins, and we found that the effect on transport upon siRNA knockdown is highly consistent with the predicted effects from the protein coloration analysis, therefore validating, reinforcing the feasibility of inferring protein regulatory mechanisms from protein variation across single cells. I'll close by a part that is very dear to my heart, and this is sharing the methodology and the data with the broader community so that we can enable the research of our collaborators, colleagues, and the community at large. We put a lot of effort in sharing not only the data, but also the metadata and the code so that our analysis can be reproduced. And we have benefited from colleagues reproducing our analysis. We also worked with other members of the community to formulate standards and benchmarks and best practice recommendations for performing single cell protein measurements that should be helpful for people who would like to start doing such analysis. And this is an ongoing effort where we solicit feedback and more experiences from people who adopt these methods so that we can update the recommendations for the second version. Everything that I had the pleasure of sharing with you has been part of a very collaborative and fun research effort, mostly in my group at Northeastern University, but also some of the data, especially on the multiplexing with mustags now at Parallel Square Technology Institute. And the last project that I mentioned, in identifying regulators of protein transport, was led by Jason Dirks, who was also the lead student for developing PlexDA in my group, and Shirley Duke developed the sample preparation method that I mentioned. Shirit Sur was the lead author for the alternate RNA decoding, the first part of my presentation. And of course, everything has been highly collaborative work benefiting from everybody in our group and generously supported by many funding sources from the NIH Directors Award, and I particularly want to acknowledge the Allen Distinguished Investigator Award by the Allen Frontiers Group. I'll stop here, and I'll be happy to take questions if we have time.
Dr. Slavov, it was a great talk. You have the floor open for any question. Feel free to unmute yourself.
There seems to be a question in the chat. Are variations distantly separated in linear sequence but close in folded protein occurring in structurally or functionally important regions of the folded proteins, or the opposite? That's a very good question that can help us generate at least a hypothesis for their functional importance. We haven't done this analysis fully systematically, but we certainly have observed such 3D spatial clustering in highly conserved functional protein domains. And therefore, our hypothesis is that this structural clustering of amino acid variation is functionally important, but that remains to be tested and confirmed based on direct measurements.
Hey, great talk. Can you hear me?
Yes.
Awesome. So I saw that you were doing some like deep learning regression tasks on the some of your data. I was just asking, have you tried any other like extended linear models or like a transformer model or anything like more like I guess like complex than a regression task, or do you just find that regression just works really good for your task?
So the context in which we did deep learning was for predicting peptide properties,
Yeah.
such as fragmentation and chromatographic separation, migration times. And these models were developed by other colleagues. We used their models. The fields have a long history of modeling with various approaches, classical approaches, long before deep learning emerged. And classical models could predict fragmentation, but their accuracy was always lower
Yes.
than empirical measurements. And the current deep learning models have accuracy that approaches empirical replicate measurements. So they're just more accurate in that context. Some of the other models that we did, for example, the model for predicting the RAS ratios, the ratios of substituted to unsubstituted proteins, those were simpler linear models and regressions. They were not deep learning models.
I beat those. Awesome, awesome. Thank you so much.
I have a quick question. So most of the data set that you have online publicly accessible is like the blood cell types, like even hematopoietic cell types. How difficult is it for like developing neuronal data sets?
How difficult would be for other people to acquire single cell mass spec proteomics data? Is that the question?
Yes.
It very much so. From my perspective, it is not difficult at all, and I would like to make it very easy, but this question needs some caveats and to be put in perspective. So for people who understand mass spec proteomics and have access to instrumentation, even if that instrumentation is not state of the art, not the latest instruments, acquiring your own data should be quite feasible and not difficult at all. I think probably the largest difficulty is conceptual and intellectual for many people. It is not so much that the facilities don't exist, but the awareness of what is possible and how to design the experiments and how to acquire the data is not there. And to help with that, we organize workshops. Some of those are hands-on workshops where they have a lab component. Of course, these workshops are harder to scale. It is difficult to have thousands of people join a lab workshop, but we try to do what we can. And the other aspect, of course, is still having some mass spec facilities and having the ability to have clean enough sample preparation, having good chromatographic separation and all of that. So even if you don't need to have the latest instrumentation, you still need to have reasonably good instruments, which I think is present in most major universities across the US, certainly R01 institutions. And of course, having the latest mass spec instruments does afford significant benefits in terms of sensitivity if you want to quantify particularly challenging lowly abundant proteins. But we started the field of single cell mass spec proteomics at a time when the community was very skeptical. There were many established people who did not believe that it's possible, which is why nobody had done it before. And we used relatively old instruments. We used instruments from 2010 to do the initial proof of concept measurements that ultimately led to changing the thinking, in the community, and to embracing this possibility in developing more methods and developing instruments that can do these measurements more sensitively. And all of that has been wonderful, but it still remains the case that those instruments from 2010 are able to acquire useful data. And this, if you allow me to go on a short digression, I think is also a good lesson for younger colleagues, for early career investigators who might feel at a disadvantage of not having resources to compete with well-powered groups. I was one of those. I started my lab without having institutional resources to purchase any instrumentation. And I convinced a good friend of mine from Harvard, Bogdan Budnik, who was manager of the mass spec facility, to collaborate to start acquiring the first measurements, and ultimately worked from there. But we had instrumentation that for its time was outdated, wasn't cutting edge, and yet we used it to accomplish something that people having better instruments had not done previously and thought not possible. So I think that for me is an example where young people with optimism and bold ideas can make real contributions, even if they are at a disadvantage in terms of the resources that they have.
That sounds good. Thank you, Dr. Slavov, for today's talk. Are there any more questions? If not, please join me in thanking Dr. Slavov for today's talk.
Thank you for the invitation and for having me.